Morning. Morning, ladies and gentlemen.
Good morning.
Y'all hanging in there? Woo, been a long week, had it? Getting nervous for this thing. I'm trying to get here a little earlier to make sure it's working, after our fun last week or Monday. I'm a little nervous for this thing. All right, here we come. Here comes everybody. We still early. We early, guys. We're doing good. 5 minutes early. What's going on? What's up? Slowly we're getting in here. Get some more folks in here. Sorry. Taking some notes, trying to get some thoughts on. Morning, everybody. Morning. All right, let's give everybody another second to roll in. We got a few minutes. Y'all did good, guys. Getting up this morning, man. Y'all did good. All right, here we go. Hey guys, we're gonna take a break. We take a break. I'm gonna print a new roll. This thing should have been finalized Tuesday. I'm looking around my desk. It's laying here somewhere. And I need to call, make sure I got everybody in the class this afternoon. So this morning to break. So we'll do that. Let's definitely take a roll call. But just kind of get through some material here. We're a little light today. We have about 20, and we have 22 people in here. Like we're taking some cuts today. Here comes a few more folk. Now they're coming in. All right. I've got around 8, so give me another second. All right. Man, I look like some tired folks. It's been a long week. Maybe things will settle in a little bit. We'll get a rhythm going here. All right. We're going to have about 5 people taking a siesta today, huh? All right. I got about 8:01, guys. We got a long way to go and a short time to get there. So I'm going to go ahead and get started real quick. Let's give them one more minute. At least my clock may not be your clock. Hey, ladies, y'all find it interesting that 2 people that don't have their camera on are 2 boys? You're so vain. You probably think this video's about you. Come on, Logan and Zach. Let's all do this, man. We're going to get the video on. Nobody looks like, there you go. Logan knows I'm harassing him. Come on, Zach. You know you're looking good this morning. Come on. All right. Here comes Molly. Molly's dragging in. Molly went to church with my mama. We're going to be late to work. We're going to be late to church. All right. Here we go. Actually a pretty interesting lecture today. I think y'all will enjoy this. I'm going to be honest with you. I think some of them, like I've already said, bones can get a little bit dry to me, but I'll try to make it interesting because there's just not a lot going on there. Nothing new being covered. But today is one of my favorite lectures in the whole world of biology. So I think we'll end on a pretty good note today, I believe. Okay, real quick, let's get rolling, guys. I appreciate y'all. Look, first of all, okay, let's start off with y'all want some good news? Don't y'all think we need good news this morning? Okay, we are 1/4 done. We are 25% through with this class. Yes. And we've already had a test, whatever. I can't even keep my mind straight. We already had a test. I have 2 tests next week. So I am very thrilled with you guys, right? We are 1/4 done. Okay, now a couple of things that some people, and actually I've had 2 or 3 students come by and want to look at their test. I told y'all, if y'all want to, we will try to let y'all, I'll let y'all look at them Monday after you finish your test. So when you turn in that other test, you can sit down and look at it, okay? So I think it's going to be the easiest way to do it because it's just hard to do it with lab and all. And I've got to run, get a bite to eat between those labs. And I don't want to be grouchy for that 1:30 lab. But we are 1/4 through, people. This is intense, rapid fire, psychosis, A&P. I don't really think you could teach this any quicker. Okay, so I'm thinking this is the absolute fastest you would ever want to take a class. Okay, is that fair enough? So that to me is pretty wild. Rapid fire A&P. All right, second note. Okay, I had a couple of people that maybe some of my guys that came in Tuesday, because Monday, I went over all the rules, but a couple of thoughts here. Now on test day, which is what, Monday, we will not have Zoom meeting, right? Right? Y'all don't have to get up at 8 o'clock and be on Zoom. We're going to meet up here in 107, what was it, 105, 106, and 104, I believe, those 3 little rooms that we all took the test in. Come down to the first floor. I believe we're getting close to everybody today. Looking good. So I got 26. We still got, I think, a couple of people out this morning. Okay, 28, but I got, okay. But so don't forget on Monday, we don't have lab Monday. We're taking tests on chapter 3, and we'll meet up here at 9 o'clock. I think that worked really good. Other than on whatever day, my days are running together. I thought it worked pretty good Wednesday, didn't y'all? I mean, we had good safe spacing, and y'all had plenty of time. If y'all get here and we start around 9, obviously we don't have anything else after that, but I'll give y'all an hour and a half, and I think that's plenty right to knock out that test. Okay. All right. Now on Tuesday, we will have Zoom at 8 o'clock. We'll start with chapter 4, which is histology, which should be a very easy chapter since we've been doing that already in lab for a day or for at least a day or two. And then we will have labs at 11 and 1:30. Okay. Now, for those in my lab, let me just remind y'all, what's our test on Tuesday? Connective tissue, epithelium, stages of mitosis, right? So we're mainly going to be on the new stuff, the connective tissue, and it'll be at the end of lab. So all the quizzes next week for my labs, Tuesday, Wednesday will be at the end of lab, and then our midterm is on Thursday. So this game goes really fast, and I'll get y'all review sheet Tuesday. Okay. So don't forget, we'll have we won't lecture Monday. We'll lecture on Tuesday. Okay. Now I think it's Megan. Megan came by and talked to me. They were talking about school and stuff. And I know some other ladies did. Several of y'all talked to me, but kind of a combination of some of the talk. But I think Megan's the one that really hit this on me that I think y'all need to think about. In a regular school year, the homework, the mastering homework, which is going to count what as a regular test grade, right? All of that, y'all know that, right? Y'all got the code in there. I normally try to put that due the day we cover it. So y'all would have had to have finished. I would have made you have chapter 3 done yesterday. But because this is so fast, and Monday, you're trying to find out, you don't even know where Canvas is at. I think it was due, is it due today or something like that? Probably due today. I'm going to give y'all a great big thing that will help you a lot. If y'all will do the mastering before I talk about that chapter, your grade will probably go up 5 to 10 to 15 points. Okay. Because here's how this works. If y'all will read the chapter, do that homework, and you try to figure it out, my job is to try to hit the hard spots, right? And regardless of my level of teaching, if y'all do the pre-lab work, I'm telling you, this gets way easy. I mean, I'm telling you, because that way, when I say a word like goji, you read it last night, you made fun of it, and it's not as scary. It doesn't intimidate you as bad, right? And so I had one of y'all even told me that, I did this, I got ahead of the day and did 3, and 3 seems to be better, than 2, because chemistry always scares me. So I know I should have said it on the first day. I'm just doing the best I can, but think about that. I think that's a really good, greatest thing I think I heard of any of us talking this week is do these mastering homeworks and, and read your chapters as best you can before we cover it. And then you can come to class with questions already written out that you wondered about, right? And so when I get to that question in the lecture, you can just stop and say, raise your hand, hold down space bar, unmute yourself, and ask me a good question. Okay, is that cool? All right, so do that mastering before. I would really do it before we cover that chapter. Okay, so that means you want to do 4 before Tuesday and 5 maybe before Tuesday, so that you'll be good about that test we got next Friday on tissues and skin. Okay, which should be the easiest test in A&P 1. So next Friday we got one on Monday, but next Friday's test will be one of the easiest ones. It's the easiest of these 6 tests, in my opinion. Okay. All right. Now y'all hang on. I'm going to try now to share my screen, and you know how good I am at this technology. I very easily can mess it up, right? All right. Let me see if this is going to work. Okay. Y'all see that one? Everybody okay? I still have trouble getting the come on, open it up. Give me a second here. All right, that's close enough. All right. Now I backed up a little bit. We actually stopped at the end of a really good break. We actually stopped. Oh, come on. My buttons don't want to work. Hang on. Give me a second here. It's going to be like, it wants to be like this. All right, here we go. So I had, y'all see that okay? Is that flicking? Okay. We actually stopped right here. But today, and this is a very tough topic, probably the toughest topic we got to cover in A&P 1 with muscle contraction. This and muscle contraction are tricky. But I think a good spot might be to back up and talk about chromatin, because we're going to be talking about that today anyway. So in the nucleus, by the way, I got lights. I left here last night about 6 o'clock, and they did not have my lights fixed. So they must have got in here real early this morning and put new lights in. So I'm really proud of them. They were the maintenance guys were cranking it up. They beat me this morning. But inside this nucleus, guys, these 46 chromosomes, 23 that you got from mama and 23 you got from daddy, are in there. And what we're going to see this morning is one of the most important things in all of biology, and that is how these genes are expressed to make everything we see, all the diversity we see on Earth. Okay? So in other words, this is the deal. Today, I'm going to do my best to show you how the key to life on planet Earth. Okay? So what we're going to see is that this DNA is going to tell us how to make proteins, and proteins make everything. Okay? That enzymes, they make enzymes that make everything in your body, including, look, we won't debate about this, but things like personality, IQ, whether you like chocolate ice cream or you're a strawberry ice cream person. I like them both. So, but anyway, everything, all these little traits of yours are going to evolve from this concept of protein synthesis. Okay. Really big. But the chromosomes, if you remember, when they unravel, as we saw them in interphase, they're called chromatin, right? And you can't see chromosomes in interphase, but we know they're there. We've looked at them with these electron microscopes, and they look like a spiraling staircase. So DNA, and by the way, that might be a good question on the test Monday. Describe the basic structure of DNA, right? Maybe draw a little bit of it. I may have to, maybe once you draw me a little piece. But DNA is a spiraling staircase, and I'll show a better picture than this in a minute. It wraps itself around little. Everybody know what I mean when I say a spool? Like a thread spool, or I used to fish a lot. You'd buy fishing line, and it comes on those spools. Okay, so God has these little protein spools called histones that the DNA coils around, and then they super coil up into these chromosomes that we talk about. And I know y'all remember this because I beat this in lab hard. A X is a duplicated chromosome held together, and I haven't covered this yet, at a structure called a centromere. Okay. Now I'm telling y'all, can we get a few more words and start with C? One more time. Unraveled DNA is chromatin. It coils up and forms a chromosome. When it's duplicated as an X, it is actually two sister chromatids held together at a structure called a centromere. Okay, now just to show how stupid this can be, when it lines up at metaphase, it is these centrioles that pull it apart. Remember the tow trucks? And two of them become a centrosome. Bam. Is this enough for C words? I know I just did 6 or 7 C words right there. So don't let the verbiage cloud the process. To me, I'd rather you understand what's going on here and really understand it. And if you miss a point on a test, I don't worry about that as much as if you see it. So chromosomes, duplicated chromosomes, have two sister chromatids held together at a centromere that are actually pulled apart in anaphase by the centrioles. Wow, lot of C words, right? Is that not crazy? So what we're going to see today is we're going to see that sequences of these nucleotides are called a gene. And a gene is a sequence of DNA that codes for one protein. Okay? So DNA has secret codes in it that tell us how to build proteins. And if you remember, the main thing that determines a protein is the sequence of amino acids in the chain. And so in this DNA, there's got to be a secret code, a secret message code to tell the cells how to make this amino acid chain, thereby making all proteins on Earth. Okay. Now I'm probably going to come back to this picture, it'll probably pop up again, but I thought that was a really good idea. That's in the nucleus. The dark spot in the nucleus is the nucleolus, where RNA is made. messenger RNA, and we're going to see some ribosomal RNA. And the ribosomes, those structures where proteins are made, are actually created in that nucleolus, and then they leave the nuclear pore and go out to the endoplasmic reticulum and form the rough endoplasmic reticulum. Okay, now all that said, kind of a setup for this, okay? So be sure you got your safety harness on, because this can get kind of weird. All right, so on our test, and I'll tell you what I'll do. I'm going to look it up. I'll look it up when we take a little break. But I believe this is a 5 or 10 point question on your test, okay? And I believe I give y'all a whole page, a blank piece of paper. Okay, so Faith knows if I give her a whole piece of paper, probably not be a good idea to answer that with one sentence, right? That would be bad. Okay, so you need to go deep, but don't write a dissertation. You know what I'm saying? But I would definitely use the paper on one piece of paper. Try to knock out at least a half a page to a page of writing. And I would do pictures too. Okay. Now here's the setup. Protein synthesis, and look at this. I mean, it says it right here. Making proteins from the DNA blueprint using RNA. Gene expression then is the production of proteins from a specific gene, and it takes 2 steps. So on your answer on Monday, you really need to have at least 2 paragraphs. They don't have to be very, they don't have to, I mean, obviously you can't be, but I'm giving you one piece of paper, one page, one side. Okay. But if you do not put these 2 words on your answer, you will not get a good score. Is that clear? Okay. And those 2 words are the 2 steps. The first process is transcription, and the second process is called translation. Okay. So protein synthesis is 2 different processes, two big parts. Okay. Like, yeah, I'm a huge football fan. Football has offense and defense. Okay. Protein synthesis has 2 parts to it. Transcription that actually leads to make something we need to do translation. So these actually do go together better than offense and defense. Those are probably a bad example. Okay. Now I want you to look at this right here. This is so dadgum important right here. This line right here. Wouldn't be a bad idea to chunk this at the top of the page to get a few points. Oh, sorry, I didn't mean to go forward. So here we go. So in the nucleus, the DNA is going to be copied into RNA, and that is called transcription. And we're going to look at each of these, okay? We're going to look at each of these as we go. Then the messenger RNA is going to leave the nucleus. Okay, now look, I told y'all I treat everybody the same. Emma, turn your camera on, girl. I'm going to catch you absent if you don't turn that camera on. Okay, I love you. You're a great student, but everybody's gonna have their camera on, and you didn't turn it on yesterday, I believe, if memory serves me right. So everybody get a camera on, okay? It's just fair to everybody. Treat everybody equal, right? Okay. Now, and I do appreciate you. You're a great student. Okay. So translation is going to be at the ribosome where we actually build the protein. Okay. Actually build the protein. Okay. Now that one is pretty tricky, and we'll look at it. You'll find it quite intriguing. Okay. And we'll talk about it. So we're going to look at that in just a minute. Let's see how many I got in here now. I'm kind of doing a virtual roll here. Okay. All right.
She just chatted that she can't see you on her phone. She's having to switch over.
Oh, okay. So she's made a note. Send me a note. Okay. That's cool. Because see, I'm an old man, and I don't think Emma would ever do this, but I have had people so we did this a little bit in the spring, and I had a couple people kind of game me. They turned it on, put a little picture up, probably went out, smoked some joints and had a drink or something, and came back and cut it off a little while. So anyway, I'm not trying to be ugly. I'm just trying to treat everybody fair, right? And I love you guys. Look, y'all just don't know how much I appreciate y'all, okay? Because it's been a rough semester. And some of y'all were here at Northwest last semester. I'm sure Ole Miss was a mess, but this whole flipping over to online was just a nightmare, okay? So basically everybody, we had to curve grades. We had cheating like you cannot believe, okay, of end of world level cheating, okay? Anyway, so back to our story. So two paragraphs, right? Transcription, which is copying the DNA gene into messenger RNA, and then that messenger RNA going to the ribosome, and we actually build the protein at the ribosome in a process called translation. Now, think about this. Now, we looked at this the other day, and as a matter of fact, I probably could get in a lot of trouble here backing up, which is always a bad idea. But let's just do it. I feel like doing this. Watch this. I'm going to go way back. This is probably the worst idea I've had all day. But remember this picture right here, where it was, this past it. How proteins are made. We looked at this picture. So today, we're going to actually see what's going on out here and here. So the DNA transcription occurs here. Then that messenger RNA is going to leave that little hole, go out to ribosome, build a protein that's put in the vesicle, that's carried to the Golgi, that then is carried to the bloodstream or somewhere else in the body to make you do that. Is that not cool? So I'm trying to give y'all a reference here that I think is really cool. So this is what I'm talking about today is really going on right there at the cellular level. Okay, really cool.
Where does the translation occur in that we just showed? Because that
okay,
made
cool.
a lot of
That's fine. Let me
sense.
back up. That's good. Good question.
That made a lot of sense what you just did right there.
Yeah, and I think that was worth going back. Thank you. I think that is worth, because I hate jumping around. But it happened on the red circles. Those are ribosomes on the little red circles.
And that's where translation occurs.
Matter of fact, well, I'm going to show it to you in a minute. There's good pictures coming up of it. But I don't think there's a picture in our lecture today that shows the whole picture here. I would normally draw this on the board, but I don't have I'm not, y'all saw how good I draw for this, but I would normally just draw this on the board somewhere. Most of my classrooms have a chalkboard or a marker board. I would draw it. In lab, I just go up there and draw it on the board. Okay. Oh, here comes Emma. Okay, here we go. Uh-oh. Thank you. Appreciate it. I know you're having technical problems. It's all good. Okay. All right, I'm getting there. I'm going back to where we started. See, when I jump around, okay, back to our chapter. Here we go. So let's start. So this is a good little picture here, slide here. So a gene then is a sequence of these nucleotides that determine the sequence of amino acids. Now, you already know that there are only 4 different nucleotides in DNA, right? So in that spiraling DNA, there's only 4 bases: adenine, thymine, guanine, and cytosine. Okay. So the biggest, I think, one of the big, and by the way, this actually happened in my lifetime. So I find this really cool to talk about this because this was within my lifetime, which is kind of scary when you think about it. I'm getting old, and I'm going to get a pen out here and try to help a little bit. But watch. But check out this map. If there are 20 amino acids that make up all proteins, right? How are we going to tell which of them 20 we want if there's only 4 letters in the language of nucleic acids, right? The 4 letters are what?
I have a question.
Yeah.
If there's only 4 nucleic acids, then what is uracil?
Okay, let me clarify that, Faith. Okay, let me strike that. That was a mistake I made. There are 4 nucleotides in DNA. Okay, uracil is a 5th nucleotide that we only see in messenger RNA. So I will show you that in just a minute, Faith, I promise. But great question, okay? Because I did say there's only 4, but technically, for what I'm trying to say, there's only 4 in DNA. Okay. So it's a great question, Faith. So what I'll see in just a minute, uracil will be in messenger RNA, and why God did that is one of the great mysteries of biology. Why God did that little other 5th nucleotide. I have a theory. I'll tell you in a minute. Don't let me not tell you why. Okay. So you're good. I know you're on top of things. So don't let me get in a tangent and not go back to that, because that is an excellent thought. Okay. Now let me give y'all a story that I think might help here, because this isn't that long of a chapter. It's a mental understanding. Okay. So we ain't got to cover like 20 tissues or something like we did yesterday, going through all them different epitheliums. Okay. So it's not this long. It's just a conceptual thought. Okay, now I know I'm getting old, but maybe y'all remember this. How many of y'all like cereal? Y'all still eat cereal? Okay. How many of y'all noticed the toys they used to put in cereal? They still do, don't they? Like they'll have all these toys, like the kids that fight over. Matter of fact, kids sometimes will get the cereal for the toy, don't even eat the cereal. Okay, well, I'm just going to go ahead and where's Sadie? Sadie's going to love this, and I was not going to pick on her day, right? Now look, if I use your name a lot, it just means that you've impressed me, or you've talked to me, and Sadie actually has called me out already on Diet Coke, right? So Sadie is one of my, already liking her in this class. She's cool. But Sadie, don't judge me, Sadie. I am addicted to Captain Crunch. I'm just going to go ahead and say it. I like me some Captain Crunch. So when I'm really down or depressed, I'll eat a bowl of pure sugar. Captain Crunch is pure sugar. And when I was a kid, one of the little toys they had, and by the way, as you get older, Sean will tell y'all this, they just recycle them damn toys every 10 years. Okay? So when a kid gets out of like, from 6 to maybe every 15 years, they just go back and get the old one. Okay, how many of y'all remember the decoder ring, the Captain Crunch secret decoder ring, and it was real cheesy. It had, here's what it was. It had all the alphabet, and you could click it, and it had a number that went for a letter. I mean, it's a brain thing. Okay, so A is 1, 2 is B, 3 is C, 4 is whatever. And if I wanted to send, Sarah a love letter, like I think she's, pretty nice looking lady and pretty nice person. So I don't want to say I love you, Sarah, right, on a piece of paper and everybody laugh at me. I'll put it on I'll decode it into some numbers and make sure Sarah has the blue ring like I got the blue ring. And then so if a teacher gets it, it's just what? A bunch of numbers? What the crap? No, but Sarah with that blue ring would turn it over and it goes, I love you, Sarah, or something. Okay, now I know Sarah. I'm not in love with Sarah. I'm just making that because she's smiling and I'm trying to wake everybody. And I try to pick on everybody before the month's over, okay? All right. So think about this. The secret code in Captain Crunch decoder ring is what? A is 1, B is 2, C is 3. There's a secret here, but it can't be 1-to-1 ratio, right? You see what I'm saying? There's only 4. As Faith noticed, there's 5 nucleotides, but there's only 4 in DNA, so we can't have A represents 1 amino acid, right? And so back in the late 70s, early 80s, when I was in college, here's what they did. They come to this realization that probably in the secret language for life, for human gene expression, every 2 letters do it. It obviously can't be 4 or 4, but because there's only 4 combinations. But what about every 2 letters? So if you took 4 and squared, that's 16. There are 16 ways to write those 4 letters 2 at a time. A little math here, okay? And math scares me. So it can't be every 2, right? So then the scientists said, well, then maybe every 3 letters is a secret code. And when you do 4 to the 3rd power, is 64. So there's 64 ways you can write A, T, C, and G 3 at a time. Okay, so for example, let me just throw it at you. AAA, TTT, GGG, CCC, CCA, CCT, CCG. You get it. I'm not going to write out 64 of them. So, and listen, guys, listen to what I love and what I hope to do. Besides, I know I've talked too much, but I'm just going to throw it out here. This is a very, very special group of people in this room, in this class. And I am totally convinced that several of y'all could really do something great. You know what I'm saying? And really go do and discover some of this stuff. Or go make a major contribution to science or change lives as a nurse, a nurse practitioner or whatever, which some of y'all, I would buy stock in this group right now, okay? Because I believe in y'all. So here's how they did this. Out in California, Sean was raised in California. The biotech companies, a lot of colleges started getting on this, and there was a race to try to figure out the secret code for DNA to make proteins. And so the people who figured it out, who all won a Nobel Prize, they took a test tube and they put everything in this test tube needed to do translation, which is what? ATP, the 20 amino acids, ribosomes, everything needed except DNA. Excuse me, except the messenger RNA. Because we knew you have to have messenger RNA, and messenger RNA is a single strand made from DNA. So they then just started making synthetic messenger RNA to try to figure out the code. So let me show you what they did. So they took a piece of messenger RNA, and let's go draw it out here, and they just put a bunch of A's on it. Okay? And I'm going to do this real simple because I can't draw real good. I'm trying to draw 6 A's. So if I draw 6A, if I put 6, and they actually did like maybe 21. If you throw that in the tube and wait, a protein should come out with 2 amino acids stuck together, and it ought to be the same protein. Should be the same amino acid, right? If our theory is right. Everybody follow? This is really kind of deep. But I'm kind of showing you how they did it because I like to know how things work. Now, that's not in the book. Okay? Now, watch. When they did this, I got to look at this next slide to tell you what it was because I forget. I've never memorized this. This is the genetic secret code for human messenger RNA. So if you made messenger RNA that had AAA, look, first base A, second base A, you get lysine. So when they did that, they discovered that 3 messenger RNA with 3 adenines on it always makes lysine. 1 down, 63 to go. Yeah, it took a couple of years. When they figured this out, and I don't think this actually has them all on it. Somebody actually counted these. I had like my face, one of my face one semester counted. I'm like, Robin, I don't think they're all in there. Okay, well, just trust me. This is close enough. Okay, I think most of them are there. Okay, this, guys, was discovered probably in the mid-'80s. And this was the origin of the genetic revolution of genetic biotechnology, which is my favorite field of biology right now. The mutilation, but the alteration of DNA to betterment of species and organisms and humans and hopefully the planet. Okay? So everybody follow me. This is so cool. Let me show you a couple of weird things that really freaked them out. They also discovered that this is always the first 3 codons. So every 3 letters is called a codon. Every messenger RNA begins with AUG. Okay? And we believe this is God's start codon. This is where you start the sentence. All proteins begin with lysine, AUG. Is that not crazy? So that is the universal start, first codon, first amino acid in proteins. Okay? Now, I have worked in a lab before for a psychopath. I was working for a guy at Ole Miss before I came to work here 29 years ago. I'm going to send in one of them lab books because I'm going to be a turd and thank him for just about ruining my life. But I overcame his ignorance. But he, sorry, I shouldn't have said that. But anyway, but he would come in. If you messed something up, you'd get cussed out. I mean, he was ugly, mean, and loud. And he'd find out whatever was sacred to you, like for me, the Memphis Grizzlies or the Bible, that he'd make fun of those. And he would make fun of that every day. Okay? So he was really a mean guy. But I say that because look what happened when they did these 3. Don't you know somebody got cussed out over these 3? Because they would repeat that, right? Like in the experiment I showed you a while ago, UAA, 15 of them should have what, 5 amino acids stuck together, right? And we know what it was then, and nothing happened. I mean, nothing. And then after repeating it, and probably me calling Sean to look at his lab and calling Nancy, Nancy's working at a lab in Chicago. Nancy, would you do this? See if you get the same thing. And after multiple people did it, somebody had the brilliant idea that God needs a period. Now, right now, half of y'all are going, I get it. I got 3 boys and a girl going, why would he have a menstrual cycle? No, not that period. The period, end of sentence. Okay, well, we'll cover that later. I'm saving that for Angel. Okay, I got to wake y'all up. I'm saving that for Angel. Okay, so this is end of sentence. All messenger RNA ends with one of those 3 stop codons. Okay, is that not cool? So we discovered this secret code, and I told y'all the other day, I ended the lecture of the day with this statement, so I'll say it again. And as soon as they did this in California, and I'm metaphorically going to tell you, I think it was one of the colleges that did this. They went to the president of the college and said, we need $100 million and we will change the world. Or $1 million. This is expensive equipment. And I'm sure that president was probably a English major or an art, or music major, which is wonderful stuff. I love all that. And he goes, but the band needs new horns. Okay. And they did not want to sponsor this work. And so they did what all great American people do, is they went to Wall Street, made their own damn company, and they're billionaires today. It's called the whole biotech, Amogen, Biogen, Genentech. Those were the origins of the biotech companies of the 80s. Okay, and they are changing the world today. They're some of the biggest pharmaceutical companies in the world today. Okay, because they harnessed the power of the DNA. And then the big question that came up immediately was, if that's the code for humans, what about bacteria? What about fungi? What about plants? What about, algae? And upon a little bit farther study, they have discovered that is the complete genetic connect code for all life on planet Earth. thus, in my opinion, proof that you're akin to every pine tree. Okay.
I have another question.
Shoot, Faith, go. Yeah.
Okay, so I'm a little confused as to why codons are in groups of 3 when on DNA they're bonded between 2, they're pairs. So
Okay,
where did
great question, Faith. And I'm going. That's another really good question. I'm going to get there. I'm going to look at that. What happens? It only reads one side, Faith.
Oh, okay.
Like, again, it only reads one side of the staircase. It's the craziest thing. And listen, there has been millions, not millions, but there've been a whole lot of papers. I mean, look. Okay, here's what I think is going to happen, guys. Look, listen. I'm having a little fun with this. There is nobody knows why it copies one side. We're not sure how it knows which side to copy. Okay. I think, normally I'm at camp this week, and I love church camp, but I've gotten really bored with Thursday night. We always do this late night devo where everybody tells all their sins, and it goes to like 4 a.m. And then sometimes the preacher tries to get all the kids to come forward and cry and be baptized. Well, I don't think you need to drag somebody to God. God will talk to you. When you're ready, you'll do what you need to do. And so I've gotten a little cynical about that, okay? You know what I mean? But I hear it. So in other words, I'm gonna make this statement. If heaven is 8-hour devo at night when I need sleep, I don't really want to be there. You know what I'm saying? I want to be in heaven. I don't think that's gonna be heaven. I like singing, but I think after about 1,000 years of singing, I'm probably ready to play golf, or read a book. So what if heaven is obviously worship the Lord, but what if God has a class on why I made DNA like this, 100-year class? I'm in. Why didn't I come down here and stop some of this stupid stuff on earth? I think that's a great question. Okay. I think this is the devil's domain down here, personally. But anyway, I think heaven's going to be like that. I think it's going to be beyond our thoughts. So, but one of the great questions there, Faith, is that each side, it only reads one side, okay? And of that strand, it reads 3 of them. 3 nucleotides on one side codes for an amino acid. But that's a great question. I really appreciate you thinking. I know you're going to make 100. I suspect you'll do well Monday, okay? Is that not crazy? Now I will go ahead because Faith's going to read this in a couple of years somewhere else. And she's going, well, Robin was wrong. There are a couple of spelling errors in some of those very weird archaeobacteria I was telling you about. Some of those really strange bacteria that can live in boiling water, that can live on ice, that are probably living on other planets. But just for 1 or 2 exceptions, this is universal for all life on Earth. So I've been making this joke for years. We will be putting gene. Now we haven't done a lot of gene placement in humans yet. But y'all know right now that all the corn you're eating right now and all the beans that you're eating, soybeans, have had genes put in them so that Roundup won't kill them. When I was your age, I would drive a tractor over a field 7 times to plant it and harvest it. I mean, we'd cut it 4 or 5 times, maybe 3 times. We'd spray chemicals on it, we'd plant it, we'd plow it, and then we'd harvest it. Today, they have no-till planting because they just go out there and plant it, they don't even cut it, and then they spray it one time. But y'all seen these big spray rigs, and they use the traplant, what is it now, Roundup, to kill everything except the Roundup altered DNA in the plant that we're trying to grow. So they drive over it maybe 3 times now. Plant it, spray it, harvest it. Cut their gas bill in half. Everybody in here has ate any bread or corn or any food in the last week has had genetically modified DNA in their body. We're going very slow in humans. So here's my prediction. You ready? This will be a little fun. Then we'll get into the meat of it. I'll do a little better. I'll get on through it. I believe before y'all die that we will probably put the chloroplast in a pig and a cow, and then we will destroy the grain industry. I mean, corn would go to, like oil did a month ago, come get it, it's free. I don't know if y'all saw that, but there was a day during the pandemic that crude Texas oil ended selling for, hey, come get this barrel and we'll give you $20 to come get it. It was free plus $20 for one day.
57.
57. But you had to have a tanker, and you had to have a tanker to put it in right then. So it was really a paper thing, but it really kind of woke a lot of people up. Guys, there's no reason why we can't put a chloroplast in an animal and all they have to do is go out and get sun and drink water and make sugar and get fat like Robin. We just got to figure out how to do that. And look, this is in my normal lecture, but especially in honor of the idiotic crap going on in our world right now, I'm going to throw this in, which is I've been doing this for 20 years. Okay. If I'm the king of genetic engineering, I'm going to cure 2 of the worst things on earth in one fatal experiment. You ready? I'm going to put chloroplast in humans, and I'm going to end starvation. All you have to do is put your swimsuit on, go out and lay out, drink some water, and you'd be fat as Robin if you fell asleep and got sunburned. And then I will eliminate starvation and racism in one magic genetic experiment. We all green now. Racism just ended. Everybody's damn green. What do you think? And starvation ended. All farmers go broke now. Okay, so now we got unemployment at 50%. It's at 20% today. I've been telling that for about 20 years. And one of my cool students walked out one day and made this comment, and I literally sat down and started laughing and crying at the same time. This student came by and said, "Robin, I think that's really cool. That's interesting. I'll give them 2 weeks, and the dark greens will be fighting the light greens. You ain't green enough, or you ain't light enough." And that's a sad analogy, but there's probably some truth to that, right? That when people get scared, they think if anything don't look like me, I get mad. But we got to get past that. But then that's cool. Now you laughing at me. Let me tell you what they've already done. I've seen cotton in the Delta that glows at night. Undoubtedly, the gene in the firefly's butt moves real easy, and I've seen cotton that glows in the dark like a firefly. I've seen that put into the skin of rats so you can see the rats glow like a firefly. Now, why did they do that? Because they could. There's no reason to do that, okay? However, I actually got pissed off about that. I don't like screwing with things without a reason. But one of the things I was told in my reading was that they were trying to think about this. This is a really cool one. They were trying to link that firefly glow to cancer, and maybe inject it into you. And then when you get out of the shower in the morning, cut the light off. And if you glow, wait, I got a cancer right here. Let me go to the doctor and have it cut off because it'll glow. Wouldn't that be crazy? Kind of a little easier way to diagnose cancer, right? If you glow at night in the bed. I'm just telling you. You guys are way smarter than me. Some of y'all go run with this. There are people working on this for 20 years, and we will blend animals. We'll blend plants. I don't like bananas. I need potassium. Sadie knows I'm not eating right. But if they would put that gene for potassium in a banana in a cocoa plant, I'm all about me some chocolate. I want the cocoa banana is what I'm looking forward to. Okay. But we are doing that in genetic engineering. Okay. So one of my favorite topics in regular biology, general and majors biology is that. You do not memorize that table. If I make you do a problem, I would give you the table, but I'm probably not going to make y'all do that. Okay, mutations are when you get a change in the DNA that alters a protein, and this is the cause of all genetic diseases. So a genetic disease is when a spelling error occurs where you put the wrong amino acid in, and that causes that cell to not work right. It can cause cancer many times, or it can just prevent something being made that you got to have. Okay, and I'm not going to talk about the death count mushroom. Y'all can read that some other time. Okay, so here is transcription. We have an enzyme in our body, in our cells, called RNA polymerase. It comes in, and another enzyme called helicase unravels the spiraling staircase. It comes in, binds at the beginning, the first codon of the gene, and then it causes a production of messenger RNA, which is a copy of the RNA, of the DNA in the RNA language. Okay, it's real easy. Watch. Here it is. There's your spiraling staircase. Look down here. It's uncoiling here. The enzyme comes in. Let me blow it up a little bit better. And look what happens. Look, here is the side not being coded, Faith. Here's the side being coded. And so look, G binds to C, T to A, G to C, C to G. And so that red is the messenger RNA, G to C, A to U. Now here's where U comes in, Faith. Messenger RNA has uracil in the place of thymine. There is no thymine in messenger RNA. Now, I know the question is, why did God do that? We may have to wait for him or her to explain that later. I would speculate that God did this because if it didn't, it would just be another, it'd just be our DNA. So this is what makes messenger RNA unique, really, right? It's single strand, but it has uracil in the place of thymine. Okay? Now, let me tell you something really crazy. I've been talking about making some money. I've been watching a couple of companies now about 15 years that, and their whole gimmick, their plan is, let's say this gene right here is being copied, this messenger RNA right here, and I'm trying to get my pen up here. It's not wanting to work. But let's say this right here, let's say this right here is a gene that causes breast cancer. Okay? They are making a piece of DNA that's the opposite of this. Okay? Right here. To plug in right here and clog the whole thing up. They call it nonsense DNA. So what let's see if y'all can see this. So if I know a gene causes cancers, if I make the opposite of it, anytime that thing opens up to be expressed, to be copied, this just sticks to it and stops it. Would that not be a most brilliant drug? Okay, real quick, and I'll probably do this several times. Here is the treatment of cancer, 150 years of medicine in like 30 seconds. Cut it out. Cut that tumor off. That was late 1800s, early 1900s. Radiate the whole area. Radiation. Radiation kills you and the cancer. We try to kill the cancer before it kills you. And then 50s and 60s, chemotherapy or poisons we put in your body to kill the cancer and you. Hopefully we kill the cancer before we kill you. Maybe this is a new treatment, because this would not make your hair fall out and make you sick, because it wouldn't bind anywhere except where the cancer gene is. And then the newest one is to harness the human immune system to attack cancer and jack up our already awesome defense system. And we'll talk about that. Miss Dickens will do that, cover that next month with y'all. But is that not the coolest thing? Just a little fun side note. So there are people working on things that clog this up when it opens. Okay? So one more time. The DNA uncoils, the enzyme RNA polymerase comes in, binds, and makes a copy of this, copying this gene right here in the DNA. This is DNA, look, into that red piece, which is messenger RNA. So messenger RNA is a copy of the DNA flipped. It's the opposite of the DNA. A to T, you know, not A to T, A to U, C to G, G to C, right? Okay, and T to A. So you have the other ones. But it also has a different sugar, because remember, RNA has ribose as a sugar. Remember, nucleotides have a sugar, a base, and a phosphate group. The sugar is ribose, okay, instead of deoxyribose in the DNA nucleotides of adenine, thymine, cytosine, and guanine. Okay, so there's a little bit more chemistry there. So it's a different sugar, but uracil is seen, and that's why if you saw the genetic code a while ago, uracil, right? You don't see there are no T's on there. So this is the genetic code of life on Earth for messenger RNA. Okay, but what would be the DNA of UUU? AAA. So the genes AAA in any gene in your body code for phenylalanine, PHE. That's phenylalanine right there. Just using that metaphor. Okay, is everybody cool? And if the wrong one binds, if one of them gets out of place, then that's a mutation that leads to possibly some change in a protein. But what if UAA got mutated to UUC? Would that hurt anybody? Would that hurt you? Not a bit. You still get what? Phenylalanine. But if UUU got changed for UCU, now you got serine, and that could be a bad thing. Or it could be Megan's beautiful blonde hair, which we don't think Adam and Eve had. Or some of that redhead some of y'all got. I think we had a redhead in here somewhere, right? So is that not kind of cool? So we think, we pretty sure Adam and Eve probably was dark or black haired, okay? Brown, black hair, that's dominant. Okay, blonde and red is very, very unique, different DNA. Okay, is that cool? Okay, so transcription then is the DNA uncoils and the enzyme RNA polymerase comes in and creates messenger RNA, single strand. You see it right here, red. That then is going to what? Leave the nucleus. There it is. Look, there's the strand after we get through with transcription, and then the thing recoils, and this coils back up. Whoop, wrap it back up into a spiraling staircase. That is going to then leave the nucleus and go out to the ribosome and tell us how to make a protein. Okay?
So before the RNA polymerase comes along, so basically the DNA decides a section of it needs to be
Opened up.
transcribed.
Yeah.
And then the transcription factor comes and binds to the section that's wanting to be copied. And then the RNA polymerase comes over that and does the final unwinding and copying.
That's correct. Yeah.
Okay.
And here, look, now look, before, and I love Faith, and she's really up on this. She's thinking, I appreciate that. Another very interesting point is we don't know how that mechanism of starting and stopping the gene has been determined yet. And when we figure that out, we'll regrow a finger. Or my daddy will be given a drug to make him grow more brain cells because of his Alzheimer's he's getting. So really, we're on the crest with some really cool stuff once we break the code, figure out how that is turned on and off. But like you said, those transcription factors are the beginning of our figuring it out. Now, I'll go ahead and tell you this right now. This is what most people think. As cool as this code I just showed you, as cool as this is, okay? And that's pretty cool, right? The secret code. Did anybody have a little feeling like, that's it? I mean, come on, has anybody ever seen programming code? That crap's crazy, right? That cray-cray. We believe in the DNA, we know there's another secret code that we have not discovered yet that turns genes on and off. And that is yet to be determined. Because there's large amounts of DNA that are never transcribed. So they have to have some function probably. And we're pretty sure that function is expression of genes. So we're desperately trying to figure out how to turn these on and off, or to start transcription and to cut it on and off. Okay? And she does a really good job here, Ms. Ammerman, showing that with the promoter sequence and all this. So it's a lot more deeper, but I'm just going to give you all a quick overview there on that. Okay? Now, watch this. If it works. Oh my gosh. I'm going to see if I can get it to work today. I've yet to figure out how. Oh, whoa. I didn't mean to do that. All right, hang on. Hang on. I'm trying to get the pin off, and I'm having a little trouble with that. All right, hang on. I tell you what, maybe just do it this way. Let me just stop the. Okay, let me just do it like this. Okay, here we go. Look, one of the things I really love about this is the videos. Let's watch this video, okay? And it's going to show you transcription, if it works.
Transcription is the process in which DNA is used as a template to produce a type of RNA called messenger RNA, or mRNA.
Can y'all hear it okay?
If you imagine our genome, that is, all the genes in our DNA, as a book of recipes in which each recipe is a gene that carries out the instructions on how to synthesize proteins, then transcription copies one of those recipes from the cookbook onto a Post-it note. The Post-it note with the copied recipe is the messenger RNA. Recall that DNA and RNA have a similar way of encoding information using a sequence of 4 different nucleotide bases. For DNA, those bases are A, C, G, and T. RNA has the same A, C, and G, but it contains the base U instead of T. DNA and mRNA are complementary to each other. This means that the nucleotide bases C and G always pair together, and A always pairs with T in DNA and U in RNA. Messenger RNA carries the same information as DNA, but transcribed in a code sequence of RNA. The key enzyme that carries out transcription by reading the DNA template and building a complementary mRNA is called RNA polymerase. The name is easy to remember because you know that the enzyme synthesizes RNA and that a polymer is a long chain of subunits, in this case, a long chain of nucleotides. Now let's focus on the 3 stages of transcription: initiation, elongation, and termination. Initiation begins when proteins called transcription factors bind to the promoter, a special sequence of nucleotides on the DNA strand that indicates the starting point for RNA synthesis. Next, RNA polymerase binds to the promoter, and a complex of enzymes, including helicase, unwinds the double helix and stabilizes the individual strands of DNA. Elongation proceeds as RNA polymerase attaches one nucleotide at a time to build an mRNA transcript complementary to the DNA template strand.
And they're made elsewhere in the
RNA
cell, you know.
polymerase can elongate the growing RNA strand at speeds faster than 60 nucleotides per second. Elongation is terminated when RNA polymerase encounters a specific terminator sequence on DNA that signals the end of the gene. At this point, enzymes catalyze the release of the mRNA transcript, and the completed mRNA transcript detaches from the RNA polymerase.
That's pretty cool. You have hundreds, if not thousands, of these going on in every cell of your body all the time. So this ain't a one thing going on. This is like cray-cray going on. Okay?
It only took Henry Ford 2,000 years to figure out that process.
Amen. Assembly line. I knew it. You know what else I saw there? Y'all probably never seen this because I'm old. Does anybody ever see one of those labelers where you snap, the letters? I don't know. I said that messenger RNA come out. First time I saw that, I went, it's like a label gun. You're making you are making a message. Okay, you're making a message. Labeling is labeling something. Okay. Here's a good one here. Let me throw this at you. And she kind of stole my recipe thing. But how many of y'all? And I'll tell this because a lot of people. I think my job today is to try, and I think Faith and several of y'all, I think Sean's got it. I know some of y'all got it. There's some smiles. Several of y'all got this evil look. I think some of us don't get the big picture here. So here's a big picture metaphor I've been using for a while. Let's pretend that all my grandparents are dead, and I was raised on a dairy farm, and we ate breakfast with my grandmother every day on the farm that my dad spent his whole life. My dad ate breakfast with his mother until she died at like 80, and he was 55, probably my age, 55 years old. And I can see it right now. I close my eyes. We still own the house where we all sat and had breakfast together. My dad and his three brothers and me and all of us that worked on the farm. And we would plan the day, and she would cook us meals. She made these biscuits, y'all, that I would give my big toe for that biscuit recipe. Because she would make like 50 of them, and then we'd take a break at 10 o'clock and come have a snack and eat cold biscuits, which are really good with cold bacon. And so let's go back to that room because I still got that house. My dad owns it, and the kitchen's still there. And let's time travel back. And we've all seen Terminator, so please overlook me here. So I've got to get naked, and I can't carry a photograph or any camera with me. So I go back to that kitchen. I hug my grandmother. I tell her about her grandchildren that she never met. But I'm here. I need the damn biscuit recipe, okay, right now, because I may be thrown back in the future real quick. What am I going to do? I'm going to take a Sharpie and write it on my arm, right? A recipe is messenger RNA. And then when I come forward back to today and I'm in that same kitchen, what am I going to do? I'm going to make some biscuits. That's translation. That's making the biscuit. You have to have the recipe to make the biscuit or the cake or whatever. It helped me. I'm just trying to make sure you're catching on to what we're doing here. We're using God's recipe to make life on earth. Okay, that is profound to me. Okay. All right. Now she puts a bunch of questions in here. By the way, that video is on the study area and mastering. Y'all can watch it. Y'all can answer the questions. Probably some mastering homework uses that video too. Very good video. Okay, we got a little glitch here. Now, after the messenger RNA is made, we discovered that there are pieces that are not going to be used to make the protein. We think they may be viruses that got put into our DNA or some bacterial. So these are called introns, and they have to be cut out. So we have enzymes called spliceosomes that come in and cut these introns out and then stick the rest of it. The purple part is the one that we need called the exon, that which needs to be expressed. So this is called RNA processing, and it occurs in the cytoplasm as that messenger RNA, or in the somewhere in there. I'm not sure where I'm in my mind today, but I believe it's in the nucleus as it gets ready to leave and go out the cytoplasm. This also caused a real big glitch in genetic engineering because when we put a chloroplast gene, when we put a gene for insulin into a plant, into a rat to make insulin, it didn't work because they don't have the enzymes to cut it out the introns. Okay. So this was a real big problem in genetic engineering. So we can't move genes as easy as we thought. But we very quickly figured out a way how to make RNA that has the introns out. The viruses, the COVID virus is a retrovirus. It injects RNA into you, messenger RNA, and then it makes the DNA by an enzyme that makes it backwards. So instead of making RNA, it makes DNA from RNA. So we just stole that for viruses. So we got that covered. Don't worry about it. We can make DNA all day long with the introns out of it. Just give me the messenger RNA. All you got to do is get the messenger RNA out of the cytoplasm. But this is kind of cool. So wouldn't that be a good term? Introns, right? That's the pieces that have to be cut out. Okay. All right, let's go on down. I'm going to move on down to the. Oh, here we go. Here we go. So now, and I hope y'all don't mind me playing this video, but I think it's really good, right? I mean, y'all paid to hear me, but this is good. So now we're going to look at the processing. Actually, I may skip this one. I'm going to skip processing. Y'all, that's pretty easy, right? Just cut the introns out. Y'all can watch that one some other time. I'm trying to get us out of here on time. Now let's go to translation. Okay. Now, and we'll watch the video on it because it's really important. So in translation, we've got to build the protein using the secret code in the messenger RNA at the ribosome. Okay. We got two players we hadn't mentioned yet. Y'all got messenger RNA down. There are two other types of RNA really important here. One of them is called ribosomal RNA and transfer RNA. Now, they don't put ribosomal RNA in here, but that's the RNA that leaves the nucleus that becomes the ribosome. So ribosomal RNA makes the ribosome where we're building the protein. But transfer RNA looks just like this, and we draw it like this. It is going to be the tool or the truck or whatever, the tow truck that carries the amino acid to the ribosome to build the protein. There are 64 different of these. Each of them carry a specific amino acid based on the genetic code that we already discovered, that God set this up. And so they have an anticodon that binds to the codon. Okay, now I'm going to show you this. It's pretty darn cool. Okay, so inside, here we go. So inside the ribosome, there are 3 binding sites. AUG is the first codon. What can bind to it? And what is the only thing that will bind to it? UAC. And that's the start codon. And it always brings methionine in. Okay, what's the next one? GGC. What's the only thing that can bind to it? CCG. And that's going to bring us the specific amino acid based on the genetic code. And so all translation is these things come in until they hit it, and they build this chain until they hit a stop codon. Look at that. Bam. And it just slides right down the messenger RNA like a scanner. All right, let's let her tell it. Then I'll try to put it in a better word. Check it out.
After transcription and RNA processing are complete, the resulting messenger RNA contains the coded instructions necessary for building a specific protein. During translation, the ribosome synthesizes a chain of amino acids called a polypeptide using the mRNA transcript, much as a cook bakes a cake based on a certain recipe.
She stole my biscuit recipe.
Just as ingredients in a recipe have to be added in a certain order, so too amino acids need to be added to the growing polypeptide chain in a specific order. The order is determined by the messenger RNA, which is read in codons. Each codon consists of a series of 3 nucleotides and spells out the position of one specific amino acid. The table you are looking at shows each mRNA codon and the corresponding amino acid it specifies. Thus, the codon AUG encodes for the amino acid methionine, GGC encodes for glycine, and so on. Recall that mRNA is transcribed from DNA. So, ultimately, what determines the amino acid sequence is the DNA sequence. This genetic code is universal and is found in all living organisms. A second type of RNA important in translation is called transfer RNA or tRNA. tRNAs provide amino acids to the ribosome to incorporate into the growing polypeptide chain. There are many different tRNAs, and each carries a specific amino acid on one side and has a specific anticodon on the opposite side. The anticodon contains a sequence of 3 nucleotides and is complementary to an mRNA codon. The last important player in translation is the ribosome. We can go back to our cooking analogy. The mRNA is the recipe on the Post-it note. The tRNA is the assistant that brings the ingredients to the cook, and the ribosome is the cook.
Our oven.
The ribosome consists of 2 subunits, one large and one small. During translation, the ribosome holds the mRNA in place between the 2 subunits while providing a docking or binding site for tRNA and facilitating the codon anticodon.
Y'all watch this protein be made. Watch the protein be
The
built.
large subunit has 3 binding sites for tRNA molecules called A, P, and E. Now, let's look at the complete process of translation. Like transcription, translation occurs in 3 stages: initiation, elongation, and termination. The first step, initiation, begins when the mRNA that has left the nucleus and an initiator tRNA carrying the amino acid methionine bind to the small subunit of the ribosome. The initiator tRNA is complementary to the codon
See, AUG binds
on
the TAC
mRNA,
every time.
which is also known as the start codon. Initiation of translation is complete when the large ribosomal subunit binds to the small subunit in such a way that the mRNA is sandwiched between the 2 and the tRNA is in the P site. During the second step of translation, elongation, another tRNA binds to the ribosome.
Watch this. This is the tricky part. Watch this.
The only tRNA that can bind there is the one that has the anticodon complementary to the mRNA codon. In this way, the mRNA sequence determines the specific amino acid sequence.
Watch that protein build.
Next, the tRNA in the P site transfers its amino acid
Whole
to
thing
the
slides
tRNA
down.
in the A site.
There
And
you
a
go.
peptide bond forms between the 2 amino acids. Now the ribosome shifts down to the next codon. The shifting of the ribosome results in the empty tRNA moving to the E site, and then it exits the ribosome.
It go gets another
The
methionine.
tRNA carrying the growing polypeptide is now in the P site. Then a new tRNA arrives at the A site, and the process is repeated as the tRNA in the P site transfers its now 2 amino acids to the tRNA in the A site. The ribosome moves again. The empty
Is this
tRNA
not cool?
exits,
Look at this.
and a new tRNA arrives.
All the way to
Each
stop.
elongation
Codon.
cycle adds 1 more amino acid to the growing polypeptide. Finally, translation is terminated when the ribosome encounters a stop codon. The stop codon binds to a protein called the release factor, which releases the completed polypeptide chain from the tRNA in the P site. The polypeptide is then modified and folded into its proper 3-dimensional shape, at which point we call it a protein.
Okay, now let's think about that. So I got some DNA from my mom and daddy that was transcribed into a messenger RNA. It went to my ribosome, made a protein, right? And y'all just saw it be translated. It got folded, put in the vesicle, carried to the Golgi apparatus, shipped out into my bloodstream, and it went to my face and made me ugly. Miss Brownlee got a better strip of DNA. It went and did the same thing and made her beautiful. Life is not fair, people. Okay, right? I mean, isn't that cool? Okay. Julian is smarter than Robin because he got a better DNA sequence. Now he may not work as hard as Robin, right, or whatever. He may work, you know what I mean. But we all have little differences. This is how all that's done. Craziest thing ever. This is the way it happens. What do you think? I know we had a good run here. This is a tough one. There's not a more difficult hour to understand probably in the whole course than this one. So if you got it, you're in great shape. What do y'all think? Anybody got Faith? You got a question, Faith? She's asked. Okay, go. I love it, Faith. Go.
I just want to make sure I understand. So you have the ribosome. and you have the mRNA come along with the instructions for building, essentially.
It goes
So
to
you want to
it,
build.
and then it reads it like a recipe on making a cookie.
Yep, and so then you have, you know, the mRNA and the ribosome, and then you have the tRNA come and they bring the bricks to build the building.
Yeah.
And so then they come and they read the instructions, and as they're reading the instructions, they build their part of the building.
Let me
When that
interrupt you,
part's
Kate. Look,
done, they
here's
read.
what it does. Look, it's almost even cooler than reading. The only one that can bind here is that one. Since
Mhm.
it really has no choice, it has to bind that one. And the only
Okay.
one that can carry is glycine based on the genetic code. So in other words, God made the code, and this is just the completion of the code.
Mhm.
Saying
So
it's already all been done like that. It's just the way it is. Yeah. Isn't that cool? So then they go back and get another one. So as soon as that one leaves, it'll go get another methionine. You follow me? So they're always being recycled. These are little tow trucks bringing the amino acids down to the ribosome.
mhm.
But they only give it up when they can bind to the DNA code, to the messenger RNA that was determined by the DNA, which is the gene.
Okay.
Yeah, you're right. Everything you said is correct. I was just going to say it really doesn't read it as much as it is what it is. It's like it's like this. Think about keys. I mean, y'all can all go out in the parking lot and stick a key in there, but the only one's going to open my door is my key.
Mhm.
Right? So that's the only one that can bind, and then it passes it over like this, like right here. Glycine binds this, and it just keeps going down the line till it hits the stop codon. And then that protein then is processed by the Golgi and sent out to the cell, to the body to do whatever. Now here's the thing, and we'll take a break right now. But think about this, guys. Everything, and we can argue about this. It hasn't been determined how much is genetics in our life as how much is environment, how life unfolded. Okay? Probably one of the great mysteries of mankind. Right now, we believe that about 55 to 60% about everything that happens in your life has already been predetermined by DNA. In other words, at conception, Robinson was eliminated by the NBA. Even though I wanted to be an NBA, I want to be a Grizzly, I don't have the raw genetic material to play professional basketball. Okay? Now look, my wife taught kindergarten, and I'm going to do a little bit of preaching here for just a second. We'll take a break. But we've got to, somewhere around the 3rd or 4th grade, start getting real with these kids, right? Everybody can't be president. We ain't have it 40 what? What is Trump, 47, 48? 40, whatever he is. Do the math. There's like 250 million, what do we got? 300 million Americans. Everybody can't be a doctor. You just ain't smart enough, okay? Everybody can't drive a school bus, right? Blind people can't drive a school bus until Elon Musk makes a self-driving bus. That is the reality. Now, my wife spent her career lying to kindergartners, telling them everybody can be everything, anything they want to be. We have got to stop lying to kids. Here's what I would say. Y'all can be more than you can imagine, okay? But there are limits to what everybody can do. I can't fly. I probably would be a terrible pilot. But so early on in your life, y'all got to figure out what God-given talent you got and then try to be the best at that. And if you do that, you will be a successful person in this life. I don't care if you're scraping cow manure, which is what I used to do in a dairy farm, be the best poop scraper in the world, and the people will knock your door down to get to you. We make fun of plumbers. Do you know what a plumber makes in New York City? $150,000 a year. Then rich people pay a lot of money for somebody to pump poop for them. Okay, so never make fun of a man or woman doing her job, right? And sometimes I'll get into class and I might see Miss Jasmine at McDonald's, and I've had a few times I go through the drive-through window, and as Sadie knows, I probably go through the drive-through window too many times, and that little girl will look at me like she's embarrassed, and I will say, "You be damn proud of what you're doing. I'm proud of you. You're working. You're contributing. You're paying your own way. You're my hero. You be proud. Serve them fries. Be proud. But you make it the best damn fry you can make. Do your best. Do your best." I mean, I believe in that. Okay. But I do think, though, that most people, as a matter of fact, my heroes are people who used 100% of what they got. Okay. I think Elon Musk is one of the great visionaries of America. He's crazy, but he's a visionary. I like Forrest Gump. Forrest Gump exceeded his potential. Agree? You know what I mean? So be the best. I had a little girl, I had a guy in class one time, had been a bad car wreck. He had lost probably a fourth of his brain. He made a C, and I've never been more proud of a grade in my life at A&P. Because he got 108% out of what he had. Okay? Pretty cool. Okay, let's take a 5-minute break, and then we'll finish. We're still in good shape. Then we'll do cell cycle that we've already talked about in lab. Okay, so everybody take a break real quick. Take a break. This is a big one. That was a tough lecture for y'all. I hope y'all got it. Y'all feel good about it, Barry? No, I got thumbs up. Okay, okay. Hey, it gets easier every time I teach it, but it is a very crazy cool topic. All right, let's take a break. Take a quick break. What's up, bud? I hear you in there. Yeah, take a quick break. I'm gonna call Roe here in a minute. I've been slacking on that. All right, Julie, is everybody seeing Julian? Is Julian Gray in here today? I saw Julian. Went here. Oh, Julian, I got, okay, I know. I'm having brain freeze here, trying to do row real quick while y'all here. Jasmine Harris? Jasmine. There's Jasmine, okay. Okay, I just helped him, sorry. Before we get started, Molly Harris. Is Molly here? Maybe I'm missing Molly today. I've worked my way down here that I'm starting to have brain freeze. Michelle, I saw Michelle. Is she here? Yep, Michelle's here. Yep. Okay, William. Yep, I saw William. Okay, I got it. Kaylin Jones. Kaylin Jones. Okay. Zachariah. Yeah, Zachariah's here. Mallory. Ada. Yep, Ada's here. McCarthy. Michaela McCarthy.
Ada's here. I'm sorry.
I got you. Okay, I'm just doing a call and roll real quick before I get started back. How about McCarthy? Michaela? I don't see her name up here. I don't know if I got everybody on the screen. I may have a few. Well, when we get back, I'm sure they'll recognize. If you get an A, you're absent here. Okay, McKinney, I saw McKinney. Yep, she's here. Heather Morgan. I need to do better calling the row. Here's Heather. Heather's here. She just had a break. Emma, we've been here. Jasmine. Okay, Jasmine's here. Okay, got it. Thank you, Jasmine. Olivia. Oh yeah, Olivia's here. And Logan. Picked on Logan. I thought we talked to him. Is Logan? Man, when I look up here, it's hard as, oh yeah, Logan stepped away. Logan's here. Talbert.
I'm here.
Okay, there you are, right in front of me, Talbert. I'm sorry. I swear I'm looking at you, and yeah, sometimes the names flicker off. Sadie, of course, Sadie's here. I've been picking on Sadie. And Nancy, I thought I picked on Nancy. So is everybody about back now? It's about time. That's been about 5 minutes, hasn't it? Yeah, I've had time to go to the bathroom. Okay, help me out here. I'm double checking the row. I've got Cheney. I didn't see Cheney here. And she's one of our Cheney is the one that added, is supposed to be taking the test today. She may have left to come to get here. I'll double check with her when she gets here. I guess if she takes her test, she'll be counted here. I've got is Molly Harris here? You may be here, Molly. Are you here? Molly? Because I've got 28 people in class. I've got Molly not here, and then how about McCarthy? Michaela McCarthy. I don't see Michaela. Is she here on another screen? Is Michaela McCarthy? I don't see her. So I got 25 of y'all. Okay, I'm gonna go with that. I got McKinney's here. Everybody else I think I found here. So I got 3 not here. McCarthy.
You didn't call my name.
I didn't call a bunch of y'all at the first. I was looking at I got yeah, look up here. If you see an A, if you got a P, you're present. So I was just kind of started and I got offline. I'm just going. So I got Megan and Simone, and at the top of the alphabet, I kind of was working my way up to y'all. So here's the 3 that I've got not here. I just didn't call it. I was trying to do it while y'all were breaking, by looking at y'all's names. I see you on the screen. So I've got Cheney, Molly Harris, and Michaela McCarthy, the 3 not here. Okay, does that sound right? Everybody else good, right? Everybody else here. Okay, so if your name is one of those 3, you need to speak up. Y'all can tell I've done a real good job. I wrote down some of it, but I'm pretty much not a big fan of attendance. So we're jumping around. Let me do my attendance real quick. So I'm not going to count y'all absent until today, for Monday. I checked it Monday, but if you were not in class Monday, you're not absent. Let me finish my attendance here real quick for this first week. Okay, so this last little part is something we've already did. It is the sales cycle. Oh, could y'all see that screen then? Okay, I think it paused on me. Oh, I did. Y'all couldn't see that. Never mind. I was on a grade screen. Sorry. You have to tell me when something don't make sense. I was showing y'all. I was actually doing attendance. Here's attendance. Look, y'all couldn't see that. So everybody, I've got Cheney and the three that will not say here, Molly and Michaela. Everybody else is here. So sorry. I mean, y'all were asking. So I was just letting y'all look at the attendance sheet. I don't think there's anything unethical about y'all looking at today's attendance sheet. But I do one of these for every day. Okay. And so what I'm saying is, there's the week we've done. Look, we're already a fourth way through. And I pretty much, if you skipped Wednesday, Merry Christmas. Okay. I count you here. Okay. All right. Let's go back to, see if I can go back to this. Can y'all see protein synthesis now? Yay, nay?
It's still on translation.
Okay, cool. Thank you. Thank you. All right. Before we dive into this last part, anybody have a question on that? And it is pretty wild. I'm really going to try to make sure we get out on time. So don't worry about, y'all can watch this video on your own, but I just want to show those two main ones. Because I mean, I don't need y'all to pay me to watch y'all play a video for y'all, but I do think video, and I do think that is one of the great evolutions of education, right? Could you imagine me trying to show translation verbally without that animation of it coming in and filling in? So that does make it a whole lot better. Claudia with me. I appreciate that, Claudia, because it really has made it a lot cooler. So animation has been a big deal. This is just showing what happens after translation, how the proteins are carried off to the Golgi and all that kind of stuff, and it's got to be folded right. And y'all can check that out on your own. Okay, and here's a concept boost to showing how that works. I mean, that's pretty cool. And here I forgot, this is at the very end. So here is a gene that made Robin ugly. It was transcribed in the messenger RNA. The introns were cut out. In the nucleus, it left the nucleus and goes out to the ribosome to build a protein that becomes an enzyme that makes Robin ugly. Very cool. So everything that is a genetic nature, and we still debate about how much is genetics. And oh, by the way, just a little fun note. I didn't pull that out of the air. The 55 to 60% is based on a 30, 40-year study of identical twins separated at birth. So the perfect test group to find out if genetics or environment is more important is looking at identical twins raised in completely different families and doing an analysis of how their life turns out, what they end up doing. And we think 60% is pretty much in the genes. And 40% is, I had great parents, they treated me right. I probably turned out better than I would have been if my dad beat me every morning. I actually had a boy in my class one time told me that his dad woke him up every morning by pouring a cup of coffee on his face or on him. It wasn't boiling hot, but he would do that to him to wake him up and say, son, world's a tough thing, be tough. He got about 14 and beat his dad to death with a bat. He didn't kill him, but his dad didn't pour no coffee on him no more. So the environment has a huge effect on us too, right? So try to create a positive environment. That would go a long way. Still summarizing it, there it is. Good, great summary. The big pictures are really good. There's a big picture animation online. So check it out.
I have a question.
Yes.
Where's the ribosome, like once it goes to it? Is that already on the rough endoplastic reticulum, or is it just floating around?
Well, that's a good question. It's both, Logan. It's both. Good question. It can be on the endoplasmic reticulum, but in bacteria that don't have endoplasmic reticulum, remember they have ribosomes, and they can be free floating and they can be attached to the ribosome. They're both. Very good question. Great question. Who makes the ribosome? I don't know. Okay, I mean, but probably but honest to God, there's an enzyme that makes what? Ribosomes, right? Now, which came first, the chicken or egg? I don't know. Okay. But now remember, listen, who do you get your organelles from? I told y'all yesterday. Mama, who did she get them from? Her mama. So who did all of our organelles come from? Eve. If you believe in Adam and Eve, there's your mama. There's your organelles. So well, maybe God will tell us how he made that first set of ribosomes, okay? First cell, you know. But that is fascinating, isn't it? Crazy fascinating. Okay, now, we did this one yesterday, so this should go pretty good. We got about 40 minutes or so. So let's see if we can knock this out. Good question. Anybody else? Good question, Logan. Very good. Anybody else? Hey, I'll tell you what, too. I know it's kind of hard to do in that room, but if you do if I'll get here a little early before 9. So if you have a question, something really bugs you, you know what I mean? I don't mind going over something else to a group of y'all. I'll get here, try to get here about 8:30, 8:45, okay? Then again, you know me, I run late. So I'll try to get here about 8:45, and that way, if you have a question. But once we're testing, I can't really answer a question, right? That wouldn't be fair. Okay, so one of the great theories of biology, and remember, theory is a concept that's been proven over and over again by experiments. Okay, an hypothesis, you remember, is an educated guess, like the sun will rise tomorrow. And if we catch it rising, I've been watching it rise now for about 55 years. I think that's a theory now. It's going to come tomorrow, right? It's going to rise tomorrow. One of the fundamental theories of biology is one that I have a lot of fun with. It's called the cell theory. And what it says is that all living things are made out of cells, and they come from another living cell. So one of the pillows, or one of the cornerstones of biology, is that life only beget life. So here's the $1 million question. How'd that first cell came about? We don't know. There you go. If I were starting over, I might have went into that, because in that field, you can just make up stuff and try to prove it. And you can get published by coming up with some theory, okay, on there, some hypothesis. But anyway, and I guess this theory says this. We violated it one time when God created life, I guess. It was violated one time, or maybe God is life and he just dropped some down here. I don't know. But I can tell you this. This says, as you drive to come here Monday to take this test, if you're driving out I-55, you look over and the Energizer bunny is sitting next to you, you need to just go on up to Lakeside. Just skip the test. You go on and get yourself committed. You're going crazy. Life is not going to pop up on the floor and all of a sudden become an animal. Okay? You know what I'm saying? Life has to have life to start it. Except our one exception, maybe when life started on Earth. Okay? That's to be determined. Okay? So the cell cycle then, we talked about, and I know some of y'all are not in my lab, so I'm going to do a little bit, but I'm not going to lecture quite as much as I normally would. But when I took this years ago, it was all about those stages, right? When I took this in high school, it was all about prophase is so cool, metaphase is so cool, anaphase is so cool, telophase is so cool, interphase, and never mind, it's the resting stage, right? And I'm going to jump around and show you. But look, real quick, y'all remember these from high school? Look, you remember this? Look. Prophase, the chromosomes coil up, right? Nuclear envelope disappears. Here's all the things that happens in prophase. Then metaphase, they line up in a straight line. And that line could be any angle. It doesn't have to be left and right like this. And then in anaphase, the chromosomes pull apart, right? And then in telophase, the nuclear envelope reappears, the chromosomes unravel back to chromatin. And then cytokinesis, which is division of the cytoplasm, occurs at the same time. So you end up with two daughter cells, each with the same number of chromosomes, because remember, X's are duplicated chromosomes, and each cell gets one central chromatid, and each of those represent one unduplicated chromosome. Okay? And we just taught that forever, and it's wonderful. Then there's your two daughter cells in interphase. So when these things start to coil up, let me see if I can get the pen to come up. I may be messing up today. Again, each cell has 46 duplicated chromosomes. When we get to anaphase, the chromosomes are pulled apart. Right? And each cell gets one of these copies, which is a complete chromosome. It's just not duplicated. So in the whole process of mitosis, you have 46 chromosomes that are duplicated when we go into prophase. And through prophase, metaphase, and anaphase, we end up with two cells with 24 46 unduplicated chromosomes in it. Now, remember, I made fun about this in lab because this drove me crazy. You can't take $46 and give two people $46. The key is that X is a duplicated chromosome, and that half came over here, and this other half on that side ends up over here. That really confused me when I saw this first time in college. Does everybody understand what we're doing here? Is everybody with me? So heads up or whatever. So in cell division, nuclear division, in prophase, the unraveled chromatin coils up and forms an X. And in anaphase, we separate the sister chromatids held together in a central mirror. And that centriole has protein filaments that pull the chromosomes apart, and they end up in two separate cells. And when I saw that first time in college, I mean, the teacher went crazy, drawing prophase and metaphase and anaphase, woo, telophase. And then when he got through, he's like, and then they're in interphase. You can't see nothing. It's resting. I was taught in high school that interphase is the resting stage. And it is the resting from dividing. But I've got a test question Monday that's going to ask you 3 things that happens in interphase. And you better already, as most of y'all already know, and I know Mr. Phillip covered it too. But what we're going to see is once we discovered what I'm going to show you, we went back and rewrote it, and now we call it the cell cycle, right? And so now we do it this. The cell cycle is interphase, which starts here and goes all the way to here. And it is divided into a G1, an S, and a G2 phase. Now, cells spend about 90 or 95% of their life in interphase in either G1, S, or G2. The 3 things that happen in interphase are what? Cells grow, they get fatter, they make more organelles, they make more cytoplasm, they replicate their DNA, and then they make more. That's number 2. And then number 3 is they do their what? Cells do their job in interphase. So if God wants that cell to be storing fat in Robin's stomach so that he can skip a meal one day, maybe go camping, or when the zombie apocalypse hits, he cannot eat for 6 weeks, a month, probably 6 months, and all he needs is water. Brain cells in your head are in interphase right now trying to figure this out. Okay? So on the test, Monday, I may say, describe the cell cycle. You want to draw this circle and label it as you see here. Okay? If I ask you what happens in interphase, I would be thrilled if you told me that the cells grow, they copy their DNA, and they do their job. Okay? And then the M phase is the part of the cell cycle where we do mitosis. So prophase, metaphase, anaphase, telophase occur here, and then cytokinesis occurs during while telophase is. All of that is the M phase, which is a very short part of your life. So we made a big deal when I took this as a kid about these stages of mitosis, which represent about maybe 5% of your life, of a cell's life. The bigger mystery was what happened in interphase, but we didn't discover that until we got electron microscopes and went in and studied those organelles and saw transcription and translation occurring at the molecular level. Matter of fact, about 1988, I was at Ole Miss, and that's the first time they ever had a molecular biology class. The books, I mean, it literally was discovered in the 80s, all that protein synthesis and stuff. And I remember I've got like edition 1 of a book called, the first book ever called, molecular biology. Okay. And of course, this redefined genetics now, because this is a big deal in genetics, right? And we can now predict a lot of things with this, with genetics. Okay, I'm going to give y'all, since y'all been so nice, I'm going to give y'all another little tidbit. I'm making out this test question a couple years ago, and I said, describe the cell cycle. And I made a terrible mistake, and I give y'all a clue. Hint, it's 4 stages, or 4 parts. What do you think 50% of students draw at this point? Prophase, metaphase, anaphase, and telophase, and they lose 5 points on the test. I did not do that to be tricky. I saw cell cycle as what? G1, G2, excuse me, G1S, G2, and the M phase. Didn't say nothing about drawing those stages, right? Okay, is this not crazy? All right, my phone is blowing up. I'm gonna make them wait till we get through the class. Sorry, I cut it off. All right, so if I ask you to describe the cell cycle, draw that, and then explain it with words, okay? And then we have a short answer question on the 3 things that happen during interphase. Okay. All right, now let's see what else we got here. Talks about when all that DNA replication occurs during interphase. It's just talking, and there it is, reviewing that. And so what we're going to see now is we're going to actually take a little bit here and talk about how DNA replicates itself. I think I will play this video because one of the coolest things is DNA has to copy itself during interphase, okay? Do I have 2 videos here? Let me say that there used to be one in here, somebody scratching their leg or something. Okay, never mind. Now this is this is a little bit deeper than you need to know for this class, but let's just look at it because they spent a lot of money on these videos that I have to cut this off. Hang on. There it is.
Prior to cell division, the genetic information which is stored in chromosomes must be copied in a process called DNA replication, and the duplicated DNA separated evenly between 2 cells. Understanding of DNA replication comes largely from studies of E. coli, bacteria that are found by the billions in the large intestine. Let's take a look at how DNA replication occurs in an E. coli cell. As we zoom in, we see the DNA. At the origin of replication, the 2 strands of DNA separate and then serve as templates for making new strands. The result is a replication bubble. The bubble grows in both directions, forming 2 replication forks. In humans, numerous replication bubbles are formed. Let's zoom in on one of them. Some of the proteins that work together at the replication fork are shown here. Here, DNA helicase unwinds the 2 strands, and DNA polymerases type 3 build new strands of DNA. A critical component of the DNA polymerase 3 protein is the DNA clamp protein, which prevents the polymerase from dissociating from the template strand. Thus, the process of DNA replication is called semi-conservative. Each new double helix contains 1 original DNA strand and 1 new DNA strand. Because strands in a DNA double helix run in opposite directions, the new strands must be made in different ways. One new strand, the leading strand, is built continuously and forms from its 5' end to its 3' end.
A little bit of chemistry different than I want you to know.
The new strand, the lagging strand, is opposite to the working orientation of DNA polymerase 3, and thus the lagging strand must be built in fragments, called Okazaki fragments, in the 5' to 3' direction using multiple primers. First, let's focus on the leading strand. DNA polymerase 3 builds a new strand of DNA by adding DNA nucleotides one at a time. Each new nucleotide must pair up with its complementary nucleotide on the parental strand. Adding new nucleotides works the same way on both the leading and lagging strands. Because of the opposite orientation, replication of the lagging strand is more complicated.
Now, this is more deeper than I want y'all to know. I'm not going to make you describe this. Just don't, but just see it. I just want you to see it.
Reads the DNA strand and produces a short segment of RNA, which serves as a primer.
Enough of that. That's a little deeper. I knew that's a little deeper than I wanted to go. All right, here's our stages. Okay, let's just run through them real quick. Interphase, right? The chromosomes are unraveled and the genes are being transcribed and translated to make proteins, right? You see a nucleus, and we're at 2900 power here, but we can see this at 400 power in lab. The centrioles duplicate, and they're the tow truck that's going to pull the chromosomes apart during cell division. Now, as we enter into M stage, there are checkpoints where molecular proteins must be present for this to occur. So prophase will not start until the cell has the proper signal to do it. And here you can see the chromosomes starting to coil up, the nuclear envelope disappears, and the centrioles move to opposite ends, and they're going to build this apparatus to pull the chromosomes apart. Some books do early and late prophase. This would be early. Okay, and then we get a little bit longer to be later. And then the chromosomes line up on an equator with the centrioles here to pull the chromosomes apart. Anaphase, we pull them apart at the sister chromatids. Sister chromatids are pulled apart at the centromere, and each side gets one copy of the duplicated chromosome, and that's the math trick. You have 46 copies. When you pull them apart, everybody gets one unduplicated chromosome, so we don't have to have any magic. There's no pixel dust. There's no Disney magic in the cell. We can't just go, make it happen. Everything has to abide by the rules of physics and chemistry and math. Okay, unlike my wife's banking account, right? Okay, I always have to have the autograph for my wife. Okay, overdraft protection. All right, telophase. Then the nuclear envelope reappears, the chromosomes unravel, and then really in animals, a protein filament squeezes in and you get cleavage furrows. This would be like pulling my belt till I pull Robin into two, right? So it literally forces the division of the cytoplasm. Do not try that at home with your little brother, okay? But in plants, remember, they just put a cell wall up and then expanded the walls later. So telophase is the last part of mitosis, okay? And you end up with two daughter cells, each with 46 unduplicated chromosomes that now will copy in interphase. So they replicate there. Okay. Now I want to take a stop here, and I'm going to try to draw this. And please don't make fun of me. But just bear with me, maybe a better word. There is another nuclear division that we do, and I'm going to mention it now so that you'll know it. It won't be a new word when you get to A&P2 in the chapter on reproduction. But in our ovaries and testicles, guys and girls, we do another nuclear division called meiosis, where I've got to get the pen up here. But let me just mention it because I think it's on the test, so I should mention it. So what we do is we have 46 duplicated chromosomes, right? 2 of each type from mama, one from mama, one from daddy, okay? Now watch this. I'm just going to draw it over this picture. This is really cool. The 23 pairs pair up. The 2 number 1s come together, the 2 number 2s come together, the 2 number 3s come together, all the way down to the sex pair, the 23rd pair, okay? Now watch. This cell then is going to undergo a division. I'm going to spread it out a little bit, where each cell is going to get one of each of the pair, right? Check this out. So each cell gets one of mama's number 1 or daddy's number 1, okay? And the other cell gets the other one, okay? Now this is really cool because we don't know which one's going to go which way. This is called random alignment at metaphase 1. We're going to have a prophase 1, metaphase 1, anaphase 1, and then a telophase, and then you get this. This scrambling gives you over 7 trillion combinations of sperm and egg any one person can make that will never be the same. There are 7 trillion ways you can put 23 pairs into groups of 23. You ever had somebody in your family that looked just like mama? Like a grandchild that looks like mama? She got more grandmama's chromosomes than everybody else. The most you could get would be what? 23 from a grandparent. It's a 1 in 7 billion chance. It's crazy math. This is called meiosis 1. And now watch. Meiosis 2 is going to be basically mitosis. We're going to do another prophase, metaphase, anaphase, and telophase, except this time each cell gets a sister chromatid. Okay? So you're going to have what? 23 unduplicated chromosomes. Everybody with me? And this has got to do the same thing. So you end up with 4 cells, each with what? 23 chromosomes. Okay, now watch this. When you make a baby, daddy's got to give the baby 23 chromosomes. Mama gives it 23, so it has the right number. If you get too many, I got one bad example for you. Down syndrome, 47 chromosomes, an extra 23. Okay? Actually, it's extra number 21. It's 321 chromosomes instead of 2. Now, here's something that a lot of people don't talk about. You cannot live without 2 of all the first 22 pairs. So if you end up with 1, 1, 1, 2, 1, 3, you're a miscarriage. 1 in 5 pregnancies end in a miscarriage. Almost all of that is chromosome problems. Okay? Only one human species person, there's only one combination that lives with 45. Would you like to know what that is? This is a woman that has a disorder where she only has one X. And she lives a normal life span. I'm drawing a blank. Anybody know what it's called? I'm having a mental breakdown right now. I'm trying to remember what it's called. In my 31 years or 34 years of teaching, I have had 2 women raise their hand in that lecture and tell the room that they have this disorder, where they are the only people, really only humans alive with 45 chromosomes. Okay.
Turner syndrome.
Thank you. There you go, Heather. I was drawing a blank. So I had 2 girls with Turner. Very good. You get a bonus, but you don't need it. You're a great student. You're a good student. Hey, y'all, one girl was born at birth. The doctor thought something was wrong with her because she had an unusual look, and they did a karyotype, and she was discovered at birth to have Turner's 45. The other little girl was from a poor family, and she was 18 before she was diagnosed. She had never had a period. And they finally took her to a doctor. The other girl had had estrogen shots her whole life. And I don't want to ever characterize anybody, but these are extremely short women, usually under 5 foot tall. They have no neck. They have no sexual characteristics, very little breast development, but no adipose on the hips. And every one we've ever studied is sterile. They don't have a complete reproductive system. Now, I mean, that don't mean that won't happen someday. Somebody will, but there's never been one reported. Now you go, Rob is picking on girls. Now let me tell you how powerful the X is. If you have a baby that conceives with 45 chromosomes and happens to be a Y, that baby's dead. No one's ever been born and lived more than a day or 2 with just a Y. So in a room full of women I'm talking to, what do you think is more important, X or Y? You got to have an X to live. Y, boys, we're just a side chain. We're like another book once described it like this, and I think it's kind of funny. The female pathway is the dominant pathway. Men are the side pathway. Now, I'm not going to argue with the Bible. God made man first, but woman was second. Maybe it was the improvement. Or maybe he messed up so bad he never tried again. That's your own opinion. You make your own decision. But you're going to be a girl. Everybody's going to be a girl without that Y chromosome as an outshoot. Here's a better way. Women, y'all are Microsoft. Y'all are DOS. Y'all run the world. You know, you're Internet Explorer, and we're Apple. We're just cooler. We're kind of the little company, you know, that now, well, Apple's now bigger than them. But if you, this is a bad example. But isn't it kind of cool? Meiosis is making sperm and egg so that you can come together and have the correct number. Okay. This is said to be diploid up here when you have 2 of each type. Down here, these are haploid. And plants are doing this. Everybody's doing this. This is sexual reproduction. And that's why everybody's different. So all of these in boys would be different. I'm putting tails on these sperm. Okay. Those are sperm now. Ready to go fertilize an egg. Really fascinating to me. So you need to be aware. So meiosis is 2 divisions. Mitosis is just 1. Okay. Now that's in the chapter on reproduction coming up later, but I think it's mentioned in the reading. Okay. I'm not going to bore y'all with this. Y'all want to watch it? What are we doing on time? Y'all just gonna, okay, I got 10 minutes. Y'all read and watch these videos. That was actually the one I wanted to show y'all. I showed you the wrong one. That actually shows mitosis in real time. Okay. Y'all go watch it. If you're having trouble with mitosis, this is animation. Like I was telling Claudia was smiling at earlier. Animation really makes biology easier to teach, right? So y'all can watch that one on your own, but it's just going to show you what we drew on the board. It's going to show you all 5 stages. Okay. There are a lot of drugs that we use to stop cell division. I told my class, my lab, and I'll back up to this again. And I guess I hate I don't have everybody in lab, but I'm so glad we got a big crowd. But guys, look, cancer is uncontrolled cell cycle. That's all cancer is. Uncontrolled spinning. If we can find out how to start and stop this, we will cure cancer. Okay. And I told my labs that I think we will cure certain types of cancer in your lifetime. Hopefully in me and Sean's lifetime. Okay. And then the other one is we will regrow body parts by turning the brain back on. Everybody in here grew a kidney in the wound. There's no reason we can't find the chemicals that trigger that gene expression and regrow a kidney for a kid that needs it. Or maybe wait, more important to me, an old person that needs it. So this is one of the most important areas of research right now. They've already won the Nobel Prize by figuring out these checkpoints that cells stop at certain places in the cell cycle at the beginning of prophase and at the end of prophase, that chemicals have to be present or that cell will not grow again. And like I said in lab, y'all making no brain cells now, you make no heart cells after 21. And so if you have a heart attack, those heart cells that are killed that's it, game over. You got to live with a less than normal heart the rest of your life. Now that don't mean you can't go to the gym and get in shape, right? But you can never, right now, we cannot regrow the heart. Okay. Okay. Here are some chemicals that are very dangerous that stop cell division. Y'all may be familiar with this one. This is a group this is a real famous group of breast cancer drugs that found on a plant that stop cell division. I've used colchicine's another one that will stop the microtubules from forming, and it is used to treat gout. Okay. So a lot of things target cell division, obviously, would be really smooth. This is GO. That's the phase that when cells leave the cycle and stay that way till you die. So brain cells are all in GO if you're over 21. Your heart cells are in GO. You're not making no kidney cells. You ain't making no liver cells. So I think a big holy quest is, in biology, is to figure out how to put cells back in the loop. Very fascinating to me. Okay. And it's showing you that here's some of those checkpoints. It talks about those there. Those checkpoints where these little chemicals have to be broken off. Another thing, one of y'all, some of y'all really have had some good classes before. Another thing that's mentioned in the book, I don't think it's in the PowerPoint, but let me show you this. At the end of the chromosome, there's a little like a thread stuck on my shirt that's kind of a runner. Well, there's a piece of DNA, single strand, that falls off the chromosome. Okay, it's hanging off the edge, and these are called telomeres. Okay, telomeres. And every time a cell divides, part of that telomere falls off. And it gets shorter and shorter and shorter until the cell dies. It can't divide again. I've been watching this little company now for several years called Geron. I believe the stock went up to $2 last week. Or so anyway, I've seen it go from 2 to 25 twice. They have all the patents on this. And they are making drugs to try to stop telomeres, because cancer cells have telomeres that keep growing. The enzymes keep adding more telomeres. So if we can cut the telomeres off, we can stop cancer. Now, I don't know about y'all, but I would like to live to be 150 years old if I could have my body as it is right now. I do not want to be like that person at the nursing home. If my last 40 years is going to be like that, I'm good to go at 80. But what if we could take Megan or Miss Brownlee and put a chemical in that made them add telomeres to all their cells where they could stay nice 20-year-old for, like, let's say, maybe you look just like this for 50 years. That would be really cool, wouldn't it? And you would sell your soul for that pill, people. Do you realize if we make that pill that you would pay probably $1,000 a month to get that pill? Like another mortgage. Well, Geron has taken human cells, y'all, that divide 20 times in a lifetime. They have added telomeres to them in the laboratory and make them divide like 75 times. Okay? So I make a joke here. The fountain of youth is not going to be no river down in Florida. The fountain of youth may be some chemical that adds telomeres to your chromosomes so that you live to be 150, 160. which I believe the Bible had people living old, and I kind of believe the Bible, and I think people did live that old back then. And I think they had, shall we call it, virginal DNA that had never been altered and beaten up by nature. And we're about we may be 100, we may be 100,000 years since that first DNA, or maybe 1,000,000 years. Who knows how long it's been? So our DNA has a lot of errors in it that maybe we can clean out, and maybe we can add telomeres and make people live longer. I would pay a lot of money to get some chemical that could buy me 30, 40 more years. Most people say the human body could live to be 160, 70. If we could stop heart attacks and cancer, maybe follow Sadie's advice and eat properly. I'm being honest, but wouldn't that be really cool if we could? But this is really cool to me. These checkpoints and telomeres may be a big deal in the future. Growth factors have been discovered. They already found some of these. We have regenerated some body parts with this. Apoptosis is a term that's called programmed cell death. And this is something that really scares me. Most everybody in this video right now has genes that are going to cause them to have cancer maybe when they turn 30 or 80 or 50. And we're finding these genes every day. Like y'all heard of some of these genes that are very strongly correlated with, let's say, breast cancer, right? There's those CCR genes or whatever. There's a couple of genes. If you have 5 of these, there's like a 99% chance you get breast cancer before you turn 70. Okay, crazy numbers. Okay, maybe even before you turn 50 or 60. Apoptosis is controlled cell death, and cells are programmed to die. Let me show you one. It's that. Watch my hand. And I can't see myself up here. Let me see, so I won't look completely stupid. Let me get me on here. So when you were growing your hand in the womb, chemicals released to make this die so that the fingers keep growing. That was apoptosis. Those cells were killed so the fingers would come out. I had a friend of mine that had a webbed hand. He had a tough life. His did not properly do apoptosis, and he had webbed hands. Might have helped him swim better if he was a swimmer. So I'm just saying, but it was hard in K through 5 or 6. That's rough. Kindergartners are rough, man. They're mean. But this is called suicide cells, and they have to do this. And so tumors lose their control of cell division. So cancer is uncontrolled cell division. What we don't want is when you go to the doctor with that tumor, you want it to be benign. I actually have one of my best friends in Northwest who told me Monday that they found a tumor under their ribs, and it was hurting them. It was about the size of a grape sticking out. They could feel it under their skin, stuck to their ribs. They went to the doctor, and the doctor told them they think it's a tumor, but it's benign. They think it's benign because it moves a lot. And so they're going to have this taken out in a couple of days. And we'll know in a month or a couple of weeks if it was benign. But the doctor felt real good about it. They're going to do it outpatient. But anytime you feel a knot in your body, that's serious stuff, right? And so cancer is uncontrolled cell division. And if it spreads to other organs or into the blood, it's said to be malignant. So if you ever get malignant tumor, then that means it's spreading and it's metastasizing. So these are very important terms because everybody in this room will deal with cancer at some point in their life. Mama, daddy, unless you just get killed in a car wreck, Dave, but cardiovascular disease and cancer are the number 1, number 2 killers of people on planet Earth. And so if we can figure out cell cycle, we will have a big tool to stop cancer. Okay, and here there's kidney cancer. Look at that. I had a friend of mine that had this, and they removed half her kidney. Actually, they went in and cut this half out just like that. And now she's been about 3 years with no cancer. If I had that, you know what I'd do? I'd just cut the whole thing out. I don't want to get it out of my body completely, right? But I'm just saying, but you always have to play the statistics when you get cancer. My dad had colon cancer about 20 years ago, and they removed his part of his colon, and I had to go with him to the oncologist. And the oncologist we sat down, and he said, if we don't do anything else, Mr. Robinson, but cut it out, which he'd already had it cut out, you have a 50% chance of being alive in 5 years. And my dad really freaked out. And he didn't understand that. He thought it was like skin cancer, no big deal. And so and when we walked out there, I said, what do you think I came today, Dad? I said, this could kill you. And then the doctor said, we're going to give you a chemotherapy drug developed in 1950 and an anti-parasitic worm pill developed in like 65. And for some reason, we don't know why, but that puts you at an 85% chance of curing your cancer. Or you could try some experimental biotech drug that we don't know is going to work or not. And so we had to sit down and make a hard decision. And you know what we did? We rolled the dice with the 85%. I'll take 85% chance every time. And I don't want to be a guinea pig for my dad. I didn't want him to be a guinea pig and try some drug that don't work. But it's just been proven, right? Like I said, he has been fine. He has no colon cancer. It's 20 years later. He's 78. Anyway, that's a true story. All right. Questions, anybody? Thoughts about the test? I really appreciate y'all paying attention today. I really do. I can look at that. I really appreciate y'all giving me an ugly look, or Claudia smiling, or Julian giving me a thumbs up. It lets me know what's going on. Yes, Miss Brownlee. Yes, ma'am.
Okay, so the test is just on chapter 3, nothing to do with the cells from lab.
Chapter 3.
Okay.
So and that's a long chapter, right? It's the only test in A&P 1 or 2. This one chapter.
And we don't.
Chapter 3 is too long.
And just mitosis, not the meiosis that we talked about, right?
That would make it. It could be a bonus.
Okay, gotcha.
But if it is, it would be more than 2 or 3 points. And it could be, since I mentioned it. It is in the book. But it's not I would never make that a 10-point question. You know what I'm saying? That could be a term. I don't think it's on the test, but it could be a term. That would have been fair. And you just say, making sperm and egg, new cell division, making sperm and egg. But I wanted you to know that term because it's a big one. You got to know it later in A&P 2. Good question, though. Good question, Miss Brownlee. Miss Brownlow. Sorry, Miss Brownlow. Say your first. Simone. Simone. I like that name. I'm just terrible with names. I'm sorry. I will call you Miss Brownlow as respectfully because I don't want to mess your first name up. You've been really good. I like it. You've been really asking good questions. Anybody else? Good question. And Faith has been rolling out with some good questions too. Anybody else?
This doesn't, it's more just a personal thing, but when you're talking about the meiosis and so on and so forth and the importance of it, my wife and I had 2 miscarriages and it was because of a lack of cell division. It was the cells got there, and I'm trying to remember what the name of it was. They called it, it skips me right now, but
she was probably not doing that. It probably wasn't that one of y'all were not doing that second division, and you probably had one like the egg or sperm had 23, another had 46.
It was a group of cells. It never did split and do what it was supposed to do, and so you wonder why this is important.
I would say this too, Shane. I'm glad you said that, Sean, because it's happened to us too. We had our first girl that's 28. Then we had another pregnancy 2 years later, and we went about 5 months, which is 6 months. That's tough. And we lost that baby. And I actually took to the doctor because I wanted to give her estrogen and progesterone to not lose the baby, right? And my best friend said, kind of blew my mind. He goes, "Do you really want to do that?" And I go, "What do you mean?" He says, "Well, something's wrong in development, and your body, maybe God is saying it ain't meant to be." And so we did not do that treatment, and she lost the baby. And it really put her in a real depression for a long time, as I know you dealt with too. And hey, listen, this is real, but you know what really helped her more than anything is when she went to a group of ladies meeting, really a church group, but it could be any kind of support group, could be anything. And every woman in that room had a miscarriage. If you try to have 5 or 6 kids, you will have a miscarriage. It's just that common. And and when you feel other people having that same problem, it seems to give you some degree of it ain't just me. And that helps my wife a lot. And I want you ladies to know that, because, and men especially, you need to know that, because if I could go back in time, that's one I would have handled a little bit better.
On our second one, the gynecologist explained the lack of cell divisions and what it was doing and everything, and he basically said that if it could have gone on, because we were at 3 months, he said that if it could have gone on, it would have not been a human because of the lack of cell division. They just kind of massed up in there but didn't divide like they were supposed to do. And so you wonder why this is important. That's the thing. And when you said 1 out of 5 or 20%, that's he said that so many people have them and don't know about it.
Yeah, a lot of women have gotten pregnant and then just had what they thought was a really hard period, which was a miscarriage. Okay, because that's what happens in a miscarriage. That whole endometrium layer is shed, and you have a period. Hey guys, great class today. Thank y'all so much. Y'all really inspired me. Anybody else got a question? I'll see y'all at 9 o'clock on on Monday. Y'all can email me. I'll probably respond to y'all's email over the weekend if something really driving you crazy, okay? All right, let's call it a day. Thanks. Good comments, everybody. Y'all have a good one. I'll see y'all on Monday. 1/4 done. 25% home. All right, see y'all.
Mr. Robinson.
Yeah, hang on. I'll talk to you. I'm just going to let everybody go because I got good students that don't want to leave. They think I'm through. I'm through. Y'all, well, we done. I'll hang on, Tom.
Okay.
Yeah, who's got a question?
I don't know why I'm not coming up like at the top, but can you hear me?
I got you. Yes, ma'am. You're good. Yeah, I hear you.
Okay, through the process of, for the test, you want us to know the protein synthesis. Do we need to go into great detail about the transcription and translation too?
I want you to write a small paragraph at each of those.
Okay.
Yeah, in other words, if you just write me a sentence and this, I'm going to give you like half credit. You know what I mean? If you just do like that first statement, protein synthesis is transcription and translation. That's going to get you 3 or 4 points out of 10. You know what I mean? Or 2 out of 5. But I want you to kind of give me a definition of transcription and translation. And I encourage you to use a picture, draw a little something too. And I think drawing helps a lot. Because if I see you draw it, then I know you know it. Maybe just don't put it in a verbal way, that as easily as it may be difficult for you to verbalize it. I would write it out on a piece of paper, watch TV for a while, then grab a blank piece of paper and try to rewrite it. Because really, if I'm giving you the test, which I just did, make an answer out, memorize it, give it to me back on the test.
Right.
But by Sunday afternoon, get you 5, 6 blank pieces of paper and take it 4 or 5 times till you got what you want to write.
Right.
That's what I do. Good question, Miss Mallory. Yeah. But yeah, so I want a little bit of detail. I think I'm going to give y'all a page, and I meant to look at the test. I think it's maybe a 5 or 10. It's 5 or 10 point question on the test. Matter of fact, let me look. Anybody else while I'm digging up the test? Oh, I had it laid out. Let's see what it is. Woo! 15-point question.
Okay. So go into great detail.
There you go. You get bonus for hanging around after class, right? So 15-point question, protein synthesis. So I said 5, 10 is 15. Okay, anybody else? So I'm glad you asked that because me and Megan were talking about it. She's still in the group. And I told her that's what I would do. I would write that out, make me an answer that's a page long. And when you start that test Monday, do that first. Just flip to the back and do that first. It's the last page of the test. Okay, anybody else? Question? Anybody? Appreciate you guys. Good class today. Anybody else? Okay. Now we'll get some other chapters that ain't quite as fun as this, but we'll get through them a little quicker. Heather, do you have a question? Heather?
Yeah. I was just wanting to figure out about the initiation process of the promoters. What were those for? Were they the?
Okay, and I'm not going to really. You don't have to really. I'm not going to expect you to put a lot of that down because that's really a molecular biology thing. But these are the chemicals that call that messenger RNA to come in and bind there and transcribe that gene.
Okay.
So, because in other words, that's getting into that one that we haven't figured out completely yet. That how does it know where to go to read to transcribe? Right. And so those initiation factors trigger that gene to be turned on. So those are chemicals that are made in the cell. Yeah.
So it's just really like the initiation for the mRNA to come in.
That tells it where to start.
Okay.
And copying.
Thank you.
No, it's really wild. And hey, look, that gets real deep, and I can show you a picture. It's crazy. There's 14 things got to bind for that to happen. Crazy. But no, but you're good. You're right. You're thinking about it. I'm not going to expect you to go that deep. If you just said that they're there, I'd love to cry. We're grading the paper. Man, look at her. She was here this morning. You just got to get here for the test, didn't you? Okay, we'll get you going here in just a second. Okay, everybody good? Good? That's not everybody good? All right, I'll see y'all. But email me if you have a question, okay? Have a good one. And I'll get this up. I'll load it up so we'll have all these online before I leave today.
Okay, thank you.
I forgot to do that other day. I've got it ready to tape up.